Good morning everybody. So, today we will continue with rate processes, and we were discussing kinetics of some specific reactions, and under that heading we will talk about enzyme catalyzed reactions. So, catalysis we already have talked about. It is a substance that generally what it does is it increases the rate, I mean efficiency of the process. So, in absence of catalyst, maybe some reactions are found to be very slow, or the yield of reaction, that is amount of product that is formed is very small, but if you apply catalyst, then it is found that the yield is increased, and it takes less time for the reaction to occur. So, like catalysis, so enzyme catalysis is typically a biological catalysis. Basically, enzymes are large molecules. I mean enzymes are generally proteins. So, proteins means they are composed of amino acid residues joined together by peptide bonds, and these basically large peptides, I mean proteins. So, having many number of amino acid residues, they are when they are bonded together to form the protein. So, these protein molecules have typical a 3-dimensional structure, and in that 3-dimensional structure is very specific. Okay. And these protein molecules, those which are called enzymes, are acting as biological catalysis for many reactions which are found to occur in maybe in human systems, or maybe plants, maybe animals. Okay. So, and nowadays these enzymes are used in synthesizing many chemicals, or doing many chemical processes in the industries. Okay. So, industrial applications of enzymes are huge nowadays. Now, if we start to talk with about this enzyme catalyst reactions, so three things are there. One is the enzyme, that is a biomolecule, large biomolecule. Then substrate on which this enzyme will act. Okay. So, substrate will be converted to a product. So, basically what is happening, that you have got the enzyme E, okay, and then this enzyme will with your substrate, it will give you product plus enzyme back. So, it is used again. Okay. So, it is recycled in this way. So, basically substrate to product. Okay. So, this way enzyme is entering, and that way enzyme is coming. Okay. So, plus E, this way minus E. Okay. So, enzyme is eliminated, enzyme is included. So, this is known to occur again via a complexation process, that is enzyme first reacts with your substrate, okay, and it is producing enzyme substrate complex. So, enzyme substrate complex is in equilibrium with enzyme plus substrate. So, it is your reactant side, this is your intermediate side, and this enzyme substrate will further undergo reaction, okay, to give you product plus enzyme back. So, basically this is the intermediate, and we have to maybe apply this steady state approximation onto this, okay. And the reaction rate is found to depend directly on the concentration of substrate, okay, directly on the concentration of substrate, and therefore your overall rate is proportional to E s, and so rate because this is your product formation step. So, therefore, rate is equal to some constant rate constant into E s. Okay. So, these are the two elementary, basically 3 elementary steps, 1, 2 and 3 elementary steps. Okay. Now, what are enzymes? Enzymes are proteins having molecular weight more than 10,000 gram per mole, that is 10 kilodalton, and it has got high degree of specificity. It is very specific. Okay. Specific means one enzyme will recognize a particular substrate, or maybe a group of substrates having some specific functionality, some specific functional groups are there. So, this enzyme will recognize that substrates which are having specific functional groups, and high degree of specificity, that is they will react with one substance and one substance mainly, or maybe one group of substances having specific functionalities, functional groups maybe. Now, living cells are found to contain more than or maybe around 3000 different enzymes, about 3000 different enzymes. So, just imagine a living cell, its dimension, and in that dimension there are about 3000 different enzymes. So, how complicated is the machinery, okay? And how complicated is the overall thing, that is so many processes are going on within such a small region, okay? That is, I am talking about a single cell, and you see that these reactions are now interconnected. So, if you chop off any step, then maybe functional things are impaired, cell functionality may be impaired. So, some complications may arise. So, you just imagine that about 3000 different enzymes, 3000 different biocatalysts are there. Okay. So, it is an amazing situation that we can we may be thinking of. Now, the lock and key hypothesis, previously it was hypothesized that enzymes are according to Fischer, the active site means the site where this substrate binds to the enzyme. Okay. So, that place is called the active site, where the enzyme does the chemical transformation. Okay. That is, first this enzyme substrate binds to the active site of the enzyme, and active site is rigid, and the structure of the substrate is exactly such that it fits that region. It is just like a lock and key. So, a specific key can recognize a specific lock. So, if you have 2 keys and 2 locks, so you cannot interchange the key. So, only one key will be specific for one lock, in the same way the other key will be specific for the other lock. So, it is just like a lock and key hypothesis. Okay. Now also enzymes are large molecules, as I told you that these are usually floppy molecules. Floppy molecules means they are it is changing with conditions, or maybe changing with time, changing with other factors like temperature, maybe pH, or maybe other external factors that may lead to alteration of this 3-dimensional structure. And being proteins, they are folded into a fixed configuration. It has got a definitive folded structure. Okay. It is not that suppose you have got 2 or 3 molecules of having same molecular weight and same sequence, same amino acid sequence, then 3 of them will have 3 different structures, 3 different 3-dimensional structure. It is not possible. So, it will have a very specific structure depending on the external conditions like pH, temperature, and maybe ionic strength as well. Okay. So, external factors are very important in determining what will be the overall structure of this enzyme or large protein molecules. Okay. Now, just look at this in a picture, that you have got this enzyme, it has got a specific groove, in a specific manner, and you see that this substrate is having the complementary grooves. Okay. So, it will just fit into it. Okay. It will just come and then it will bind, this substrate will bind, this substrate will bind to this enzyme. Okay. So, you see that this groove is exactly complementary to this part, and again this part is complementary to this part. Okay. Then this part is exactly complementary to this part, and so on. Okay. So, that is why you can say that when substrate binds to the enzyme, producing the enzyme substrate complex at first. So, it is basically, you see that these groups are not seen now. So, it is basically just like a overall thing at a combined stuff. Okay. Then with time, this enzyme substrate complex, what it will do, that it will do further reaction to give you enzyme back, and now products are separated. So, basically you see that the substrate was like this, and your product is that maybe from substrate a portion has been taken out, has been snatched out to give you the other part. So, basically these are the products. Okay. Maybe this basically represents a say breaking of some big molecule to small molecule, large molecule to small molecule. Large means say moderately large molecule, or maybe suppose a substrate is dissociating into two products via this enzyme. Okay. So, enzyme plus substrate producing enzyme substrate complex, then enzyme back, and then these are the these two are the products. Okay. So, it is something like lock and key, this represents some kind of lock and key picture. Okay. But nowadays this lock and key picture is no longer that popular. Okay. So, nowadays the Michaelis-Menten mechanism, okay, Michaelis-Menten mechanism is thought to be the right mechanism. Okay. So, in enzyme kinetics, it is the building block of enzyme, of understanding of enzyme kinetics. Okay. So, what is that? In enzyme kinetics, this enzyme plus substrate producing enzyme substrate complex. So, you just go back to the earlier slide, that this enzyme substrate producing enzyme substrate complex, and then it is dissociating to the product. So, you see enzyme plus substrate giving rise to enzyme substrate complex. Okay. So, that there is an equilibrium between enzyme substrate and enzyme plus substrate, with Q1 for the forward and K minus 1 for the back reaction. So, forward rate constant is Q1 for the formation of ES, and backward dissociation of ES will be K minus 1, and K2 is the product formation step. Okay. So, it is a quick equilibrium and then maybe slow dissociation to give a product plus enzyme back. Okay. So, it is nothing but the enzyme substrate complex that dissociates to give you your desired product. Okay. So, you see that this is the intermediate through which this enzyme plus substrate, or maybe substrate with the help of enzyme, is converted to product. So, that means we should apply steady state approximation to this intermediate complex, that is ES. Okay. So, we have to apply steady state condition for ES, and then we will find out the concentration of ES, and then we will use it further for finding out the overall rate of reaction, rate constant. Okay. So, when applying steady state approximation, this is the mechanism. Okay. So, note that the formation of product depends directly on ES. You see this step directly depends on this. So, this is one elementary step. Okay. So, therefore, K2 into ES concentration. So, give you that will give you the product, that is your desired product. Now, what is the rate of formation of ES? Now, rate of formation of ES is this step. I mean, this is the formation step, and this is your dissociation step, the lower the back reaction, and this is again by this route the amount of ES, that is ES concentration, is depleted. So, that these all three should be taken into consideration, while finding out the rate of formation of ES. Okay. So, therefore, the corresponding differential equation will be ddt of ES, that is rate of change of ES concentration with time. Okay. So, let us write down the the step E plus S giving rise to ES product plus enzyme back. This is K2, this is K1, this is K minus 1. So, ddt of ES is equal to K1 into E S, because it is the formation step, then minus K minus 1 into E S minus K2 into E S. Okay. This is the rate of change of ES with time, I mean time rate of development of ES. Okay. So, now you apply steady state approximation. So, you put this to be equal to 0, that means this is equal to 0. This is equal to 0 means your K2 plus K1, K2 plus sorry K minus 1, K2 plus K minus 1 into ES is equal to K1 E S, or concentration of ES intermediate K1 by K minus 1 plus K2 E S. Okay. Because you are applying steady state approximation. So, and this is your substrate concentration. So, at a given substrate concentration you can do this. So, therefore, let us put this wherever S is there, S0, initial concentration of the substrate. Okay. So, initial concentration of the substrate is given like this. Okay. So, you got the concentration of your intermediate. Now, it is time to find out the rate of formation of product in terms of this ES. So, therefore, your d dt of P, that is product formation, is equal to K2 E S. So, that is equal to your K1 K2 by K minus 1 plus K2 into E enzyme concentration into S initial concentration of S substrate concentration. Okay. Okay. Now, let us do one thing, that we have to think of the overall concentration of enzyme. So, initial concentration of enzyme is E0, which is equal to free enzyme concentration plus enzyme substrate concentration. Okay. So, this is your initial concentration, this is your free enzyme concentration, this is your enzyme substrate complex concentration. Therefore, your E will be equal to E 0 minus E s. Okay. Therefore, what we have to do is E s will be your concentration of E s, what we wrote like E s is equal to K 1 divided by K minus 1 plus K 2, then E into S 0, that you can replace this E with this one. Okay. So, that means K 1 divided by K minus 1 plus K 2, okay. Then S 0, S 0, okay. Then S 0 is there. So, E is E 0 minus E s, okay. So, that means left hand side you have E s and right hand side is also with E s, okay. So, that means if you transpose, then your E s will come out to be equal to, your E s will come out to be equal to E s will be equal to, you transpose. So, let us do it again. So, let us do transposition. So, E s will be equal to K 1 by K minus 1 plus K 2, okay. Then S 0 E 0, okay. This is one term. Another term will be minus K 1 by K minus 1 plus K 2, okay, K minus 1 plus K 2. Then it will be with a minus sign, then S 0 into E s, okay. So, that means if you transpose this to this side, then this will be your K 1 S 0, okay, K 1 S 0 divided by K minus 1 plus K 2, this one, okay. Then plus 1 times E s will be equal to K 1 by K minus 1 plus K 2 E 0 S 0, okay. This one is for this one and this one has been transported over here. So, times E 0, okay. So, that means what we got is E s will be equal to like K 1 S 0 plus K 2 plus K minus 1 divided by K 1 minus K 2, okay. So, you will be having K 1 divided by K minus 1 plus K 2 E 0 S 0 S 0, okay, times you will be getting another term that K 1 E s is there. So, that means K 1 K minus 1 plus K 2 K minus 1 plus K 2, okay, in the numerator, and in the denominator you will be having K minus 1 plus K 2 plus K 1 S 0. So, these two are cancelled. So, therefore, it will be K 1 E 0 divided by K minus 1 plus K 2 plus K 1 S naught. So, this is the enzyme substrate concentration, okay, enzyme substrate complex concentration. Okay. So, if we substitute, then your rate expression will be basically your rate expression. Let us write down the rate expression, that is rate of formation of product. So, Vp, velocity of product formation is k2. Okay. So, that means your k2 k1 divided by k minus 1 plus k2 plus k1. Okay, so this is Es. Therefore, this is k2 divided by k1. So, therefore, it will be k2 if you divide this in the numerator, if you divide, numerator is divided by k1 and also denominator is divided by q1. So, it will be k2 E0 S0 divided by your k minus 1 plus k2 divided by k1 plus S0. So, let us call, so this is basically combination of 3 rate constant terms. So, this is for the forward and these two are for the equilibrium state, that is equilibrium state of formation of equilibrium steps of formation of this Es formation and then dissociation of Es to E plus S. So, therefore, let us write this as a new, giving a new name Km plus S0, which is called the Michaelis constant, Michaelis-Menten constant. Okay, K2 E0 S0. Okay. So, this is the rate, velocity of product form, velocity of reaction, you can write in this way. Okay. So, if you plot the reaction velocity, okay, as a function of substrate concentration, then this will be, the graph will be like, you see that it will show like this. Initially it will rise and then ultimately it will be close to saturation. Okay. So, it will go to this direction, it is not shown this portion, it is still going up and saturates at some other point. So, that portion is not shown totally. So, only a little portion is shown. So, that is why V max. Okay. So, V max is this, okay, where the maximum velocity of this plot. So, this is your V max. Okay. So, it is like a nonlinear, looks like a parabola. Okay. Okay, nonlinear one. So, parabola like dependence, not exactly parabola like dependence means it is not very clear from here anyway. So, but it is like this. So, it is V max and this is your maybe V max by 2 and this is your substrate concentration. Okay. Now, what is this, why I have written V max by 2? Okay. So, okay. So, this is your V, okay. This is your Vp, Vp is equal to K2 into E0 S0 divided by Km plus S0. Okay. Now, if your substrate concentration is very high, if your substrate concentration is very high like this, then what is going to happen? That you can neglect this Km compared to this. Therefore, this equation reduces to Vp when substrate concentration limit substrate concentration S0 is very high, then this equation becomes K2 into E0. So, that means you can write in place of K2 E0 for your Michaelis-Menten equation like your Vp is equal to your K2 E0 S0 divided by Km plus your S0. So, if this is neglected, then these two will just cross out. So, this will be K2 into E0. In that case, this Vp will call as V max. Okay. So, when limit concentration of this is infinity. So, that is equal to given name V max. So, if you write this, Vp is equal to your this one V max S0 divided by Km plus S0. So, in terms of V max, it is like this. Okay. Therefore, why half height has been taken like this? So, this is your half height. Okay. Vm by 2, Vm, this is V max, this is your velocity, this is your substrate concentration. Okay. So, this axis. So, if we put V equal to V max by 2, then what happens? Let us see. So, V max by 2 is equal to V max S0 divided by Km plus S0. Okay. So, you just transpose it, that means V max S0 half plus V max Km V max into half is equal to V max S naught. Okay. So, that means if you just cross out V max from both the sides, you will be getting your Km is equal to you see that if you remove it, then this gives you S0, because if you transpose it here, it here, therefore, it becomes half V max is 0. So, V max gets cancelled. So, and this happens when with this condition, that when means your V is V max by 2, then corresponding substrate concentration is nothing but your Km. So, Km is nothing but the substrate concentration at half maximal velocity. Okay, that is why this Vm by 2 I have chosen. So, it is S, I mean this you can write as Km. So, substrate concentration at half maximal speed. So, the Michaelis constant, what is this Michaelis constant? Michaelis constant is defined as like what is that? Let us see. Km is equal to K2 plus K minus 1 by K1. Okay. So, what is this? This is you again write enzyme plus substrate, enzyme substrate, then product. Okay. K1, K minus 1, K2. Okay. So, this is basically in the numerator. So, high means when it is this is high, high means it may be that this is very high, this is very high, or maybe this could be very high, or in other words this may be slow, I mean this may be low, so that this Km becomes high. Okay. So, in that way means relative weightage of which step is is more pronounced, or which step is important, I mean whether this K1 is more, K minus 1 is more, or K2 is more. So, their relative magnitudes will determine whether Michaelis constant will be high or low. Okay. Now, the rate constant for the product formation K2 is called the turnover number for the catalyst, and ratio of K2 by Km is called the catalytic efficiency. Okay. So, these are defined terms. Okay. Now, we can come back to this slide, that when means Vmax slide, that Vmax is the maximum velocity for the reaction, and limiting value of the reaction rate at high initial substrate concentration is called the Vmax. Okay. Now, again let us do the plot of the inverse of the reaction rate versus inverse of the initial substrate concentration. If you do the double reciprocal, it is called the double reciprocal, then this graph linearizes. Okay. So, that means here you have got this rate means inverse of rate versus inverse of the initial substrate concentration will give you these numbers Km by Vmax, and in intercept will give you 1 by Vmax. Okay. So, this equation, as I told you, that it is a hyperbolic equation. Okay. That is, I told you that it is non-linear, different from parabola of course, and it is a hyperbolic equation. Okay. So, so therefore, difficult to determine Vmax experimentally. Experimentally means just finding out velocity of reaction, and it is difficult since where is this since this one, if you look into this graph, then it is asymptotic, going and going, rising and rising. So, experimentally where to where from you have to draw this line, it is difficult. So, that is why to get more information, it is always better to express this complicated equation in a linearized fashion. That is why this double reciprocal plot, that is inverse of reaction rate versus inverse of initial substrate concentration, that is plotted. And you see, if you do this two inversion plot, then then you see that it is just like a linear equation, that your y will be 1 by v and x will be 1 by s naught. So, it is easy to handle rather than doing it in a non-linear fashion. So, this is the velocity and this is your substrate concentration. So, this is not a good choice. So, therefore, we do not like this, we do not use it in general. So, you have to use this Lineweaver Bark plot. There are other plots also, but this is the celebrated expression. Okay. So, it is difficult to determine Vmax experimentally, and the equation for a hyperbola can be transformed into equation for straight line by taking the reciprocal of each side. That is why you have done this double reciprocal plot. Okay. And the formula for the straight line, it is y is equal to mx plus b. So, b is the intercept and m is the slope. So, just comparing these two, 1 by v is equal to Km by Vmax into 1 by s plus 1 by Vmax, like this. Okay. So, slope will be Km by Vmax and intercept will be 1 by Vmax. So, intercept will inverse of intercept will directly give you the V max. It is easier to deal with. So, that is 1 by V is equal to K m by V max into 1 by S naught or S initial substrate concentration plus 1 by V max. Okay. So, this is your intercept, this is your slope. Okay. So, this will give you you know easily easily you can deal with the data rather than dealing with nonlinear fashion. It is in a linear fashion and you can manage the data easily. Okay. So, this plot is known as the Lineweaver-Burke plot or double reciprocal plot. So, let us go, how does it look, this graph, how does that look? Okay. So, you see that this is your this is the plot. So, these are the experimental data, because you may not need to go to this concentration, maybe you do this concentration, slope is K m by V max, intercept is this, intercept will be giving you 1 by V max. Now, if you keep on going to the other side of 1 by S 0, then what would you get? Let us go back to the earlier slide, that this, just put your y value to 0. Okay. So, if this is 0, then what happens? Okay. Let us try that. So, we have got 1 by V is equal to K m by V max into 1 by S plus 1 by V max. So, now put this to be equal to 0. Why this to be equal to 0? Because we want to extrapolate further, so that this line touches your x axis, and under that condition y value is 0. So, that is why we are putting this to be equal to 0. So, that means this is equal to 0 means you put this equal to 0 under that condition. So, your so what you get, K m by S is equal to minus 1. Okay. So, what is happening, that your this is basically from this you write 1 by S at that condition. So, 1 by S when it is touching the x axis, that is your minus 1 by K m. So, you can directly find out K m. So, it is a nice way, it is a real nice way of finding out the individual things like 1 by V max and V max and K m, and also slope will directly give you, although slope will be giving K m by V max, but even if you do not want to find out the slope, then just keep on extrapolating until the line reaches your x axis, that is 1 by S axis, that is inverse of your substrate axis, inverse of substrate concentration axis. Okay. So, it is a nice way, it is a really nice way of dealing with enzyme substrate reaction kinetic data. Okay. So, and you have to remember that V max is nothing but the maximum velocity, okay, that is this one. When substrate concentration is very high, then your expression is simplified and the corresponding velocity is your V max. Okay, because the reaction, you see the step where in which this product is formed, that is your E s to product. So, more the substrate, more of substrate will be associated with the enzyme to produce E s, and then the more the concentration of E s, the more is the possibility for the product formation. Okay. So, that is why this is your V max, and Km is your this one, Km is K2 plus K minus 1 by K1. So, this is a combined K, means combined constant. Okay. So, what is the meaning of small k, and means small value of k, and what is the meaning of large value of k? Okay. That means if you go back to this plot, how this will this will be affected, this plot will be affected. Okay. So, 1 by k intercept. So, say 1 by k means large value of k means it will be somewhere somewhere over large value means this will be minus 1 by large means it is a smaller here this will be, since it is minus 1 by large means small means with a negative sign means it will be this way. Okay. Like 1 by 2 and 1 by 3, 1 by 3 will be here, 1 by 2 say maybe here, 1 by 3 will be somewhere it is less, still less with a minus sign. Okay. So, large Km, so this graph large or small Km will shift this point maybe this way or that way, and if Km is high, again is Km high means your slope will be high. Okay. Slope will be high means this graph will be like this. Okay. So, graph will be like this way maybe. Okay. So, Km high means your this value will be high, this value will be high means it will be coming close to this origin. Okay. So, let us again go back to the next slide, that so what is the significance of small Km? There must be significance. So, what is the significance of small Km? Now, you just again recall this expression that Km is equal to K2 plus K minus 1 by K1. Okay. So, that means this step, enzyme plus substrate giving rise to enzyme substrate giving rise to product, K1, K minus 1, K2. Okay. So, I mean small value of k, what is the meaning? Small value of k means this is K1, K1 means this step, this is large, this is large, this is large means this step is pronounced. So, back reaction is possibly less. Okay. And high Km, okay, so small Km means this is high, and high Km means this is small or this is high. In other case, small Km means this is high or this is small. This is small means your this step is also maybe less, that is why this is small, and also back reaction is less. Okay. So, you can think of the magnitude in terms of numerator and denominator. So, when denominator high means this step is stronger, I mean this is fast, I mean it is a strong bond formation, and in the same way if this is high or maybe if this is low means this may be low, if this is low means this reaction is less. So, tight binding condition, and therefore, when it is tight bound slowly it will dissociate to give you the required product. So, that is why it is a tight binding situation. And when Km is weak means Km is high means weak binding. Why? Km is high means this is high, this is low. Okay, this is low means this step is low. So, this is and this is high means this step is high and that step may be high. Okay. So, if that happens, that means easily it will dissociate to give you E plus S, or maybe it will eventually, means very fast, go to the product side. Okay. So, in this way you can think of the significance of Km. Okay. So, that means small Km means Michaelis constant is small, that means it is a tight binding, Michaelis constant high, weak binding. So, you can also rationalize in terms of this, in terms of this Line-Weaver-Bark double reciprocal plot. Okay, you can just think of what happens to this graph, I mean plot, when Km is high. Km is high means slope will be steeper, and the this minus, that is this cutting point on x axis or the 1 by s axis will be here. Okay. So, this is one point, and also suppose we have got same Km, means like suppose the situation is like this. What is the reason for this? Let us have this plot. Okay. So, we have got like this plot, maybe another plot, maybe another plot like this. So, this is your intercept. Intercept means 1 by V max. Okay. And you see intercept is same, but Km is same maximal velocity, but if with different Km. So, this is Km is more, this is less Km. So, Km, this Km, this is another Km prime, this is another Km, Km double prime. Okay. So, changing the value of Km will just and if this is remaining same, we will be seeing that it will pass through the same V max point, but this is coming close to each other. Now, in other situation what happens, say you have got like different graphs like this. This is your 1 by V and this is your 1 by S, 1 by V and 1 by S. That means your Km by V max is same, but your intercept is changing, here less, this is little more, this is more, and so as a result of which your this side is going farther. So, this is because of the slope is same, but the relative weightage of relative magnitude of Km and V max will determine, means where this this one V max is changing, although maybe Km is remaining same, slope is remaining same, Km by V max, maybe V max. So, it is slope is same, slope is remaining same means maybe this ratio is changing in such a way that slope is same. So, that means both are maybe changing, one is increasing means this is also increasing. Okay, this is also increasing means this way it is going, or maybe that way it is going. So, you can check it directly from this expression, that slope is Km by V max and intercept is 1 by V max. So, it is very difficult to conclude, means very easy to conclude the fate of this Km and V max when such graphs are obtained, such plots are obtained. So, these are a bit detailed things with respect to the Line-Weaver-Bark double reciprocal plot. Okay, so today what we learned is your enzyme kinetics, and it is means it used to be thought like the mechanism is a lock and key mechanism, but the popular thing is that nowadays it is no longer used, this lock and key idea. Now, it is thought that there is an enzyme substrate complexation, and that complex is then responsible for giving rise to giving the product, and this Km is nothing but we have just shown that Km is nothing but the substrate concentration at half maximal velocity, and this plot is this V versus, that is velocity of reaction versus substrate concentration, is a hyperbolic plot. So, this hyperbolic plot is sometimes difficult to deal with. So, therefore, we have to have a double reciprocal modification of this. So, when you do a double reciprocal plot, then this becomes a linear plot, and from that we can find out from slope, intercept, and x cutting point, we can find out the value of Km and V max. Okay. So, that is all about this enzyme catalyzed reaction. So, in our next lecture we will deal with again some specific reaction, and then we will switch on to normal catalyzed reactions in a bit detail. So, till then, have nice time. Thank you.